Module 1, Functions of the Plasma Membrane. Now that you know a bit about the structure of the plasma membrane, let's talk about what it does. Well, one major job of the membrane is to hold all the cell components inside. We can say that it delimits the cell, holding it together. The second thing it does is provide receptors so that the cell can sense what is in its environment. You can get an idea of the importance of receptors when you look at an instance when they do not work properly. Take, for example, diabetes. There are two types of diabetes. Type 1 diabetes is insulin dependent, where the person cannot make the hormone insulin. Insulin is an important chemical that signals the plasma membrane of cells to allow glucose to enter. Now, type 2 diabetes is non-insulin dependent diabetes. This is a more common condition where the receptors are not responding to the insulin in the body. The body produces plenty of insulin, but the cells either do not have enough insulin receptors or the receptors are not working properly. Therefore, glucose cannot get into cells to provide the energy they need. So receptors are key components of the plasma membrane. A third function of the plasma membrane is selective permeability. Selective permeability is the ability to let certain materials in or out while restricting others. Well, what does that mean? The membrane is a selective membrane. That means that some materials will be selected and others won't. And permeability is the ability to penetrate something. So a selectively permeable membrane selects for some materials to penetrate it while restricting entrance to other materials. How does the membrane do this? Remember how I stressed that the overall nature of the plasma membrane is lipid solubility? The membrane is made up of a majority of lipids, which makes it mostly oily. Well, polar materials dissolve polar materials. Nonpolar materials dissolve nonpolar materials. If a cell comes across a small fat molecule, the molecule will dissolve easily into the oily plasma membrane and get into the cell. So as long as the fatty molecule isn't too big, it will easily get into the cell. There are several practical applications for this feature of the membrane. When people suffer from shingles, a painful skin inflammation caused by the same virus that causes chickenpox, they often experience burning and aching pain on their skin. One treatment to help ease the pain is to use a lidocaine patch, placed directly on the skin. Lidocaine belongs to a class of drugs known as local anesthetics, and it helps to numb the pain. The patch contains the nonpolar drug. Because it is nonpolar, it can travel through the plasma membrane in the skin cells. Selective permeability also works with molecule sizes. Remember one of the proteins I introduced in the plasma membrane was a channel protein? A channel protein has a channel running through the length of it. It's big enough to allow small molecules into the cell, yet not bigger molecules. One of the more common molecules to do this is water. Water molecules need to move into and out of the cell to even out the water concentrations in the area. If there's too much water in the cell, it can easily move out through the channel proteins. Another factor that affects permeability is charge. Cells need several charged atoms and molecules, or ions, in order to operate properly. They also have to release them. If the ions are small enough, they can travel through the channel proteins, but they do this in specific ways. You see, there are many channel proteins whose amino acids form a positive or negative charge inside the channel. So if a channel has a positive charge, what types of charged molecules do you think it will attract? Yes, negatively charged ones. And if the channel has a negative charge, it will attract positive ones. So channel proteins allow the passage of small molecules in and out of the cells, as well as attract specifically charged ions. All of the selective processes I've just covered are pretty amazing ones, especially since we're just talking about the material surrounding a cell. But there's more. Cells sometimes have to take in molecules that are too big to pass through channel proteins. Not comparatively gigantic ones, but big ones like glucose. You see, glucose is a simple sugar molecule that most cells need for fuel. In fact, brain cells can only use glucose. But glucose is too big to travel through the channels of the channel proteins. So how does it get in? It utilizes carrier proteins. Carrier proteins are located in the plasma membrane and allow specific molecules into the cell via a process called mediated transport. What happens is a carrier protein is designed in a specific way so that it only accepts a molecule with a specific shape. Now, more than one molecule may have this shape, so the carrier protein may allow more than one type of molecule into the cell, as long as those molecules are the correct shape. The molecule fits into the carrier protein and they bind together. Then the carrier protein begins to change its shape. It adjusts its configuration so that the molecules can be released on the other side of the plasma membrane. This mediated transport is transport that requires a mediator, or helper, to get materials inside, and the carrier protein is the mediator. Now, there need to be 3 conditions for this process to work. The carrier protein is made for a specifically shaped molecule. We call this specificity. The carrier protein is specifically shaped to fit the molecules. It's kind of like carrying groceries home from the store in paper bags. If you see that there are jumbo watermelons on sale, you know that you cannot carry them inside the bags. The bags were just not designed to fit jumbo watermelons. The grocery store would have to provide you with large, sturdy plastic carriers specifically designed to carry the watermelons. So, if a cell needs to take in glucose, there are carrier proteins designed to accept glucose specifically. The second condition, besides specificity, is competition. Say when you're at the grocery store, they're running out of bags. They just don't have enough bags to contain all the groceries you want. But you need to put your groceries in bags to carry them to your house. So the bagger begins to put the items in closest to him, leaving the extra materials at the store. In the same way, with mediated transport, there are lots of molecules with similar shapes that may fit into the carrier protein, so they compete for them. Whatever molecules are in the highest concentration and can move the closest to the cells will find their way into carrier proteins. So I discussed specificity, competition, and now let's talk about saturation. If you really need to take all your groceries home but don't have enough bags to hold all of them, you can say the bags are saturated. They can only hold a specific amount at a time, limiting the number of groceries you can transport. In the same way, if you have a molecule that has to get into the cell via mediated transport, there's a limit to how many of that molecule can enter the cell at a time. If a carrier protein is busy transporting molecules through a membrane, it's saturated. It can't transport any more any faster. Yet, if there are only a few molecules around to bind to a carrier protein, it's not saturated. So when more molecules suddenly arrive to the cell, it could transport them quickly. Well, I just covered the several factors that lead to the selective permeability of the plasma membrane. In review, there's the lipid-soluble nature that allows fat-soluble molecules to dissolve through the membrane and into the cell. Channel proteins allow small molecules to travel through. Some channel proteins have an overall electrical charge in their channels to allow specifically charged small ions to enter the cell. And there are carrier proteins that transport slightly larger molecules through the membrane based on specific shapes and quantities of those molecules. Now, I haven't talked yet about how proteins get into the cell. Proteins are very large molecules. I've only discussed small and medium-ish sized molecules. We'll cover that in the next section.